atmosphere of Earth is overwhelmingly made of nitrogen. If aliens could see our nitrogen-dominated atmosphere, they might imagine the animals or plants of Earth breathing the nitrogen to make use of it. After all, about 78% of the air available to us is nitrogen. Nitrogen makes possible amino acids whose very name reminds us that they contain amino groups of NH2. And nitrogen also makes possible the DNA and RNA, which need 2 to 5 nitrogens for every single base. But there's a big surprise, a big obstacle. Our cells are unable to directly take up the free nitrogen from the atmosphere and use it to build amino acids and nitrogenous bases of DNA. Plants also haven't figured out the trick for getting nitrogen from the atmosphere. They're in the same boat we are. So nitrogen everywhere, but not a molecule of it we can use. Fortunately, bacteria have our backs because some of them can pull nitrogen from the atmosphere. You suppose that's why they call them bacteria? Fortunately, there are a few plants that can partner with these bacteria to help capture nitrogen. All the rest of life on Earth depends indirectly on those few microbes partnered with a select group of plants. The result is a major bottleneck in the movement of nitrogen into living cells. This is unlike any other metabolic pathway in that every other living cell is ultimately relying on nitrogen that made it through this bottleneck to arrive in a form that we can use. In this lecture, we'll examine the fascinating flow of nitrogen from bacteria to plants to us. We'll see how our own cells build important nitrogenous molecules, both amino acids and the bases of DNA and RNA. We'll also see how cells break them down and return them to be recycled. With an eye towards expanding the bottleneck, we'll examine a variety of corn that grows 16 feet tall without fertilizer, and we'll consider some intriguing implications for other plants. Let's return to nitrogen in the air. To be absorbed and used by animals or plants, nitrogen must be converted to forms of nitrogen that plants can use, either ammonia, NH3, or nitrate, NO3-. Converting nitrogen to ammonia is no easy task. As we saw much earlier, the industrial process for making ammonia from nitrogen requires 400 to 500 degrees Celsius and over 200 atmospheres of pressure. Now, fortunately, cells capture nitrogen at normal temperatures and pressure thanks to a remarkable enzyme called nitrogenase. Some of these bacteria that grab or fix nitrogen are free living in the soil or in water. Other nitrogen-fixing bacteria form close associations with the roots of certain plants. Most of the nitrogen gets fixed by bacteria called rhizobia, which colonize the roots of leguminous plants like peas, beans, and alfalfa. How the rhizobia and their legumes establish their partnership is a bit like dating. First, the plant sends signals in the soil in the form of special flavonoid attractant molecules that it secretes to lure the bacteria to its roots. The bacteria respond by attaching themselves to the root hairs of the plants and sending out their own signals called a nodulation factor or nod factor. Different species of bacteria produce slightly different nod factors, and the specific bacterial nod factor determines whether the plant will allow them to colonize the roots of the plant or not. Once this compatibility is established, bacteria move into root cells that then divide to form little swellings on the roots called nodules. And it is inside these nodules that nitrogen fixation occurs. So what is fixing nitrogen? The first thing to remember about converting nitrogen from N2 to NH3 is that it gains electrons and gets reduced. The electron source for reducing nitrogen comes from a small iron-containing protein called ferredoxin. The enzyme that catalyzes this reduction is called the nitrogenase complex. It has two components. The first funnels electrons one at a time from an iron-sulfur protein, that's the ferredoxin, to the nitrogenase. The second component catalyzes the reduction of nitrogen to ammonia by the nitrogenase. This results in each N atom forming bonds with 3 hydrogens. In aqueous environments, NH3 forms an NH4+ ammonium ion. Now this seems simple enough, but there's a wrinkle and a clue about why we don't fix nitrogen ourselves. Nitrogenases are inactivated by oxygen and need protection from it. So the root nodules produce a protein that removes oxygen by binding to it with high affinity. Do you remember any proteins that bind oxygen? Of course you do. Hemoglobin and myoglobin. Interestingly, plants called legumes make a related protein called leg hemoglobin. Not leg as in your leg or arm, but rather leg for legume. Leg hemoglobin resembles myoglobin. It has a single polypeptide chain, a heme group with iron, and it's even red, like ours. Leg hemoglobin is a joint construction project between the bacteria, which make the heme, and plant cells, which make the globin protein. Leg hemoglobin grabs and binds oxygen in the root nodules. Its affinity for oxygen is very high, about 10 times greater than that of hemoglobin. This sharply lowers the free oxygen concentration within the nodule, allowing nitrogenase to do its work to make ammonia, NH3, or the ammonium ion, NH4+, in water. Plants can use these forms of nitrogen. The ammonia made by nitrogen fixers can also be converted to other useful chemical forms like nitrite or nitrate by other bacterial species. But plants are limited by the amount of nitrogen fixed by these microbes. If there isn't enough available nitrogen, plants don't grow optimally. To counter this, farmers have used fertilizers such as manure and fish for thousands of years to increase crop yields. Since the early 20th century, ammonia synthesis using industrial techniques has allowed farmers to add nitrogen to the soil in large amounts. This has increased food production substantially, but with large energy and environmental costs. Fossil fuels, of course, are non-renewable resources, and runoff from fields fertilized with nitrogen enters waterways and lakes. This results in excessive growth of algae that deplete the water of oxygen, killing other aquatic organisms. Some also evaporates as nitrous oxide, and that's no laughing matter. Nitrous oxide is both a greenhouse gas and a leading source of ozone depletion. So it would be tremendously useful to be able to reduce fertilizer use. Might it be possible to transfer the nitrogen-fixing abilities of legumes to other crops? Well, unfortunately, the nitrogen-fixing system of legumes is complicated and not easily moved to other plants. A simpler solution may come from a strange variety of corn found in Mexico. This corn, which grows in very poor soils, has found a way to thrive, reaching over 16 feet in height. What's its secret? Well, it turns out it has thick, bright red aerial roots that grow out of the stem. And these roots are coated with clear, slimy mucus, full of bacteria that fix nitrogen. Now, compared with the nitrogen-fixing system of legumes, these aerial corn roots may be based on genes that are much easier to transfer to other cereal crops. That would allow the modified crops to host the same bacteria that fix substantial nitrogen for the giant corn. Even just transferring this ability to other corn varieties would actually reduce fertilizer use significantly. Okay, now let's turn our attention to using the fixed nitrogen. Plants and bacteria synthesize all 20 of the amino acids for making proteins, but animals, including humans, make only some of them, with the rest coming from diet. Let's take a look at how amino acids get made. Amino acids, as you know, have a characteristic structure. The alpha carbon is attached to an amino group, a carboxyl group, a hydrogen, and an R group or side chain that is different for each amino acid. To make amino acids, cells start with molecules produced in metabolic pathways. Starter molecules include alpha-ketoglutarate, oxaloacetate, and pyruvate. A simple example is the amino acid glutamate, which can be made by reacting an ammonium ion, NH4+, with alpha-ketoglutarate from the citric acid cycle. That's all it takes. Alpha-ketoglutarate plus inorganic nitrogen in the form of the ammonium ion, NH4+, gives glutamate, our first amino acid. Add a second ammonium ion to glutamate, and that becomes glutamine. Now, either glutamate or glutamine can then transfer that amine group to other molecules, making these two amino acids bridges between inorganic nitrogen and the nitrogen-containing compounds that cells make. There is no single unifying pathway for synthesis of all the amino acids. There are, however, a few commonalities called families, and that is how their synthesis is organized. The glutamate family, for example, starts from alpha-ketoglutarate of the citric acid cycle and goes to glutamate, which in turn is also the progenitor to arginine and proline. Also from the citric acid cycle, the aspartate family, which starts from oxaloacetate and goes to aspartate, gives rise to its other 5 members: asparagine, isoleucine, lysine, methionine, and threonine. The serine family comes from 3-phosphoglycerate of glycolysis. The aromatic family begins with PEP from the Big Bang step of glucose metabolism. The pyruvate family starts with, surprise, pyruvate to make alanine, isoleucine, valine, and leucine. And finally, the histidine family traces its roots to ribose 5-phosphate from the pentose phosphate pathway. The reactions leading to amino acids from their starting materials are as varied as the amino acids themselves, with some requiring 1 or 2 steps and the others needing 10 or more. Some, such as lysine, can be made on more than one pathway, but there are 2 classes of reactions I'm going to focus on here. The first reactions are transaminations, that is, the transfer of an amino group from one molecule to the other. The amine group transfers commonly involve glutamate as a donor or acceptor molecule. These reactions are catalyzed by enzymes called aminotransferases. For example, alanine can transfer its amino group to alpha-ketoglutarate, making glutamate and pyruvate. Glutamate, in turn, can transfer the amine group to another molecule. So in addition to being an amino acid, glutamate is a means to transfer nitrogen among many molecules. Aminotransferases use a coenzyme called pyridoxal phosphate that is a derivative of vitamin B6. Another reaction for amino acid synthesis is what's called one-carbon transfer. Now, true to its name, this reaction involves the movement of a single carbon from one molecule to another. Transfers depend on several forms of the folate vitamin B9. You see this type of transfer when the amino acid glycine is made from serine. In this reaction, a 1 carbon from serine is moved to folate, and glycine is what remains after that has been taken away. Together, a series of transaminations, 1 carbon transfers, and other reactions allow us to build about half of the amino acids that we need. The rest we get from food. Our bodies take a use it or lose it approach to protein. Yes, muscles are made of protein and can even be broken down during emergency, but muscle proteins are created supporting the purpose of movement, not storage, so we don't store them. We break down and excrete any dietary proteins that cannot be used right away, whether for new protein synthesis or oxidized for energy. And there's a lot to get rid of, because for any well-fed person, the intake of protein in the diet will generally exceed bodily needs. In addition, proteins in the body are always in a state of constant change. Many enzymes turn over in hours or minutes, and even skeletal muscle protein has an estimated half-life of only about 3 weeks. Cells have 2 built-in ways to recycle protein. A lysosome is an organelle that's full of enzymes capable of degrading not only proteins to individual amino acids, but also other sorts of biomolecules as well. By contrast, the only job of the organelle called the proteasome is to break down proteins. Proteins targeted for degradation in proteasomes are flagged in cells by attaching a small peptide known as ubiquitin to them. Marked in this way, old enzymes and other proteins are moved into the proteasomal chamber and broken first into peptides of 8 or fewer amino acids. These small peptides are then ultimately degraded to individual amino acids. Then some of the amino acids released by the protein degradation are reused in the synthesis of new proteins. Whene'er my proteins go kaput, if they are past their prime, the cells will act to soon replace all of my old enzymes. They know which ones to break apart. Ubiquitin's the sign, a marker for proteasomes to find the old enzymes. These soon get bound and then cut up in pieces less than 9. More chopping yields the single ones, building blocks from old enzymes. So in a way the cell knows well of father time, it's true. Amino acids, when reused, turn the old enzymes to new. Cells are pretty thrifty and salvage what they can before they get rid of anything, and amino acids are no exception. The breakdown of most amino acids is a bit like the reverse of their synthesis. Let's recall how transamination added an amino group onto pyruvate from glycolysis to make alanine. And the molecules oxaloacetate and alpha-ketoglutarate from the citric acid cycle undergo similar transaminations. Well, those same intermediate molecules are recovered during the breakdown of most amino acids and can enter into the citric acid cycle or other pathways in the cell to be used for energy production. But when amino groups are removed from amino acids, where do they go? The body must balance amines in the body because too many are toxic. Consequently, pathways for excretion are important. Transamination reactions, as I've been describing, just shuffle amino groups around. They're not lost yet. In brains, glutamate functions not only as a component of proteins, but also as a neurotransmitter, a molecule involved in signaling between individual nerve cells. In this role, glutamate gets broken down after use, and the ammonia released is highly toxic. Now, fortunately, there's a lot of glutamate in the brain, so when the ammonia gets released, it can be combined with another glutamate molecule to make glutamine. And it is in this form of glutamine that nitrogen moves out of the brain to the liver via the bloodstream because it crosses the blood-brain barrier. A different set of deamination reactions can occur in muscles, particularly during strenuous exercise or starvation. In these muscle cells, amino groups get handed off to pyruvate, making alanine. The alanine is sent through the bloodstream to the liver, where it also gives up its amino group to make glutamate and pyruvate. The liver, as you know, can use pyruvate to make glucose to send it back to the muscles. This is not a cycle inside individual cells, but a larger scale cycle operating between muscles and liver, much like the Cori cycle with lactate and glucose. This one is known as the glucose-alanine cycle, and it keeps muscles supplied with energy in times of need. The glutamate, meanwhile, joins the pool of glutamate in the liver that will be broken down. Now, in our bodies, the liver plays a major role in recycling amino acids, in addition to everything else that it does. It converts the ammonia released in their breakdown into urea, which is excreted mostly in urine, but also in trace amounts in sweat. Aquatic animals can excrete the ammonia produced in their cells directly into the water around them. But most land animals convert the ammonia into urea before excretion. Birds and reptiles have a slightly different pathway and excrete nitrogen in the less soluble uric acid. Okay, so basically the amino groups from amino acids are getting collected in the liver into a few amino acids, primarily glutamate and aspartate. What happens next? Well, as you may have gathered, a major player is glutamate, which is broken down to release ammonia, which gets mopped up by the metabolic pathway known as the urea cycle. With each turn of the urea cycle, one molecule of ammonia and one amino group from aspartate are combined with carbon dioxide to ultimately form one molecule of urea. Urea is very water soluble, and it provides an efficient means of lowering the body's load of nitrogen. The cycle encompasses 6 reactions. Intermediates include 2 amino acids found in proteins, these are aspartate and arginine, and 2 other amino acids that are not even used to make proteins. They're known as ornithine and citrulline. Urea is formed by splitting arginine, and the cycle is circular like the citric acid cycle. One urea cycle byproduct is fumarate, which we encountered in the citric acid cycle. Fumarate, released by the urea cycle, can re-enter the citric acid cycle and get oxidized to oxaloacetate, which can then be transaminated to form another aspartate that feeds back into the urea cycle. So fumarate and aspartate link together the citric acid cycle to the urea cycle. The urea cycle was worked out by Sir Hans Krebs. Yes, he of the citric acid cycle, also called the Krebs cycle. That leads to this linkage of the 2 cycles being given the tongue-in-cheek name of the Krebs bicycle. Okay. So glutamate and glutamine between them play important roles in managing nitrogen levels in our bodies, with any excess amino acids stripped of their nitrogen in the liver. The ammonia produced is converted to urea, and that gets excreted. In addition to its other roles, glutamine is also the nitrogen control point for creation of nucleotides, which are the building blocks of nucleic acids, another critical area of nitrogen metabolism. Nucleotides, let's remember, have 3 components: a sugar, 1 or more phosphates, and a nitrogenous base, which is why nitrogen is important. All nucleotides are initially synthesized as ribonucleoside monophosphates. That is, there's a base, a ribose sugar, and 1 phosphate. If the nucleotides being made are for DNA, then the ribose sugar gets converted to deoxyribose, and we'll see about that in a bit. Additional phosphates are added to make the triphosphate nucleotides needed for both DNA and for RNA. There are 2 categories of bases: purine bases that correspond to adenine or guanine, and pyrimidine bases that include cytosine, thymine, and uracil. The source of the sugar phosphate for all nucleotides is a molecule called phosphoribosyl pyrophosphate. I'll just call it PRPP. The nucleotide assembly differs depending on whether it contains a purine or pyrimidine. Purine nucleotides get assembled atop the PRPP molecule. In this pathway, cells first make a molecule called inosine monophosphate, or IMP, that later gets converted to AMP and GMP. These nucleotides can then have additional phosphates added to make the corresponding diphosphates and triphosphates that I mentioned earlier. The challenge in nucleotide metabolism is to make carefully balanced amounts of all of the nucleotides. In the lecture on enzyme regulation, we learned about feedback inhibition, where the end product of a pathway inhibits an early enzyme in the pathway. For purine biosynthesis, PRPP amidotransferase is that first enzyme. And major products near the end of the pathway are AMP and GMP. So you'd expect that when levels of these products are high, they might bind to the first enzyme in the pathway and shut it down. And they do. But the regulation is more nuanced than simply turning off the activity of the enzyme completely when either one of these molecules binds to it. Binding of both GMP and AMP is necessary to completely inhibit the enzyme. But binding of one or the other alone only partly inhibits the enzyme. Now the logic here is clever. If the levels of AMP and GMP are both high, the cell doesn't need either, and the pathway should be completely shut down. But what if the level of AMP is high but GMP is low? If the enzyme were fully inhibited only by AMP, there would be no way to make GMP. By keeping the enzyme partly active when one purine nucleotide binds, an avenue for making the other one is left open. We also see regulation of the pathway at the branch point from IMP to AMP or GMP. The enzyme that leads to synthesis of GMP from IMP is inhibited by GMP, whereas the enzyme that leads to synthesis of AMP from IMP is inhibited by AMP. These controls ensure that appropriate levels of the two purine nucleotides are made. The pyrimidine bases C, U, and T are a little simpler in structure, and they're assembled completely before they're added to PRPP. The synthesis of all three pyrimidine nucleotides begins with making UMP, which gets phosphorylated to UDP and then to UTP. CTP is made directly from UTP by the enzyme CTP synthetase. This enzyme is inhibited by high levels of CTP, ensuring that the proper balance of UTP and CTP is made. Now, reaction 2 of pyrimidine synthesis is catalyzed by an enzyme called ATCase that we discussed back in the lecture on enzyme regulation. This reaction is activated by the purine ATP, and it's inhibited by the pyrimidine CTP. High levels of the purine ATP favor synthesis of pyrimidines, while high levels of the pyrimidine CTP stop pyrimidine synthesis. This is how purines and pyrimidines are kept in balance. So far we've talked about the synthesis of ribonucleotides, the building blocks of RNA. To make DNA nucleotides, the ribose sugar in ribonucleotides must be converted to deoxyribose, which lacks an oxygen at the 2' position of the sugar. A remarkable enzyme called ribonucleotide reductase, or RNR, acts on ADP, CDP, GDP, and UDP, converting ribose in each to its respective deoxy forms to make DADP, DCDP, DGDP, and DUDP. But converting sugar is merely the enzyme's default job. It's the regulation of this RNR enzyme that ensures synthesis of the proper amounts of each deoxyribonucleotide. Now, the enzyme has 2 allosteric sites. One is called the activity site, and it activates the enzyme if ATP binds, and it inactivates the enzyme if DATP binds. A second allosteric site, the specificity site, governs whether it is ADP, CDP, UDP, or GDP that is acted on by the enzyme. In general, binding of purines by the specificity site favors synthesis of pyrimidines, and vice versa, thus equalizing the amounts of purines and pyrimidines. At this point, the deoxyribonucleotides are all still diphosphates. To make the triphosphate building blocks, an enzyme called nucleoside diphosphate kinase, or NDPK, adds a 3rd phosphate. The other difference between RNA and DNA is that DNA replaces uracil with thymine-containing nucleotides. Deoxyribonucleotides containing thymine are started with deoxyuridine-containing nucleotides by an enzyme that simply adds a methyl group to deoxyuridine nucleotide, converting its uracil base to a thymine. Now, what about the breakdown of nucleotides? Pyrimidines are simpler. They get broken down straight to ammonia, and ammonia is removed as urea, as you have seen. Purine catabolism produces a less soluble molecule called uric acid that is normally excreted by the kidneys. If its levels in the blood are high, uric acid can form crystals that may accumulate near joints, causing a painful condition called gout. Uric acid can be excreted as it is, or it can be converted into urea for excretion. Most metabolic pathways are substantially identical in all organisms, but the nitrogen cycle involving fixation from the atmosphere is a big exception. It's fascinating and maybe a bit scary how connected all of life really is. Life on Earth depends, of course, on the energy provided by the sun and the building of atmospheric carbon dioxide into sugars by plants. But in addition, without the contributions of a handful of tiny soil microbes that convert gaseous nitrogen into forms that can be used to make amino acids, nucleotides, and other biomolecules, complex life as we know it would not be here. It's no wonder then that organisms carefully manage and recycle reduced nitrogen resources. Surely it would be wise for us to do our part to learn more about how to protect and care for the soil habitats of these important organisms.